But we are pretty proud to present Professor Sylvain Veillet, who studied applied physics in France, and then he obtained his PhD summa cum laude in physics from the University of Paris-Saclay, and then he has held positions in France, where he worked at La Petite-Salpêtrière as the head of the brain imaging group at the Cognitive Neuroscience and Brain Imaging. And then he moved to the US, where he worked as associate professor of neurology and biophysics at the Medical College of Wisconsin, and then became the founding director of the MEG program in Madison. Finally, he joined at the McGill University at the MNI in 2011, where he had been the founding director of the MEG unit, and also he was director of the McConnell Brain Imaging Center. He's also a heavy promoter of open science. And also thank you, Dr. Casani, who prepared the presentation for us.
Thank you, Rever, for the kind introduction. Very happy to be here. And I'm not really sure how this works. I guess this is the Q&A. So we are happy to provide the A's if you provide the Q's.
Please, Jose, you can start.
Thank you. Thank you both for being here with us. It's such an honor to be with you. I have a couple of questions about the whole method of this electromagnetic source imaging. So the first one is about the relevance or the importance in co-registering the electrodes. In your view, is there a better way to do it? I mean, a digital way to do it, as scanning every electrode, or a manual, I mean, to really just compute each electrode manually, or is it better to do it in the MRI with the actual electrodes?
Okay, thank you for your question. So ideally, you want to indeed have the exact location of the electrodes on the scalp of the patient. And for this, you need to indeed use a form of digitizer, right, to register the XYZ coordinates of each electrode with respect to the head. In the lab here, we use a product from the Polymus company, which has been around for many, many years. It's an electromagnetic kind of pen that lets you point at the location of the head with respect to reference, systems that you can attach both to the chair and we use like glass frames that we put on the face of the patient. And there is also a little device from that Polymus digitizer attached to the glass frame, so that we know where the head is located with respect to the room, which is fixed, of course, and therefore where the electrodes are with respect to everything else. So we scan the electrodes using this device. We looked at other solutions, like a 3D scanner, that would be much faster and less prone to human, errors. But unfortunately, it works only on people with not too much hair. So that's very limited, because once the electrode is actually, really taped or glued or even simply attached to the scalp, it gets, lost in the hair. And therefore, it's not visible to anything that uses, visible light. So we looked even at, at some point, the Xbox, the gaming console had a bar that you would attach below your TV, and there is like a ultrasound kind of system that can localize objects and bodies in the room. And we explored that solution to actually scan the head of the participants, but it didn't work out really well. I'm maybe not aware of the latest technology that could help in that respect. So I guess my short answer is that if you could use the 3D digitizer with the pen, you would not only get the electrodes location, but also the scalp fiducials that you need to register to the MRI for exact source mapping, or as exact as possible. And also to capture the shape of the head with, the pen can switch in the continuous mode of scanning, so that it basically registered the 3D points as you move the pen without clicking every time. The clicking option is useful for electrodes, but if you want to capture the shape of the head, you can go continuous mode. So that's the ideal situation. If you do have access to this, to the individual MRI of your patient, that's as much information as you can integrate into and add to the EEG for the most exact or accurate source mapping procedure. But I understand that, and we know that not every center has access to a digitizer or even an MRI, depending on, where you are. So for that, we also have tools that let you transform an MRI template so that it fits the 3D digitization of the head of your patient. And if you don't have the 3D digitization, then you can use an MR template and an EEG template. So sorry, that's a long answer, but I hope it addressed your question.
Yes, totally. Thank you. And I have a second one, if it's okay for the audience. About these distributed models, we so far have been working with S-Loretta, as the tutorial showed, but it gives you a distributed source. So in the case we would like to work with some like more specific, or like a dipole or a spot, is there a way to have a like the orientation dipole, like in the cortex, or if it's a tangential, or if it's a horizontal?
Okay, I see. So from the distributed model from S-Loretta or any other alternative in Brainstorm, there is a way to actually extract the orientation with a post-processing, some form of post-processing of the source map, but it's a bit involved. I mean, it requires that you develop your own, if you will, script that lets you extract the orientation from the source map. And this we probably can help you with, but offline, if you use the user forum. But if the question is whether Brainstorm also has, the single dipole fitting techniques that we've seen a lot in the literature, maybe less so today, but definitely in the early days of EEG source mapping. And it has some virtues and some, advantages of simplifying, if you will, the modeling of the EEG with only one dipole that maybe travels with time, And if you look at, for instance, at spikes in your patients, and you just fit dipoles on multiple spikes, etc., maybe if things go well, you can find clusters. And of course, with these dipole models, the orientation is made more explicit, and you can actually even visualize it and even quantify it so that you can assess whether it's more radial or tangential. And yes, again, the short answer is that Brainstorm has dipole fitting tools. Raimundo, maybe if you have the opportunity, you could paste the link to the tutorial sections that report on or that explain the dipole fitting in Brainstorm, and you could paste that in the chat if you don't mind, please.
Yeah, absolutely. Just to add in the those are also in the video. So when there's the source reconstruction section for the video, we only use distribute model, but also like in the tutorial that it's the line tutorial related to that data, it's the also we show how to do the dipole scanning and dipole fitting.
Oh, okay, very good.
Okay, thank you. And I have a last one, if it's okay. In the literature, there's a lot of information. Most of the good ones are from HD ESI, I mean, high resolution or high density RIs. So in the clinic, it's very difficult to have like 256 electrodes in a patient. And this is more so if we have to have a prolonged prolonged record. So there's also literature about they call it low density arrays, would have they may have 25 electrodes, some of them with the 1010 systems. In your view, from the, let's say, clinical perspective, is there a good way to do it, a standardized way to do it?
You mean source mapping with less electrodes?
Yes, I mean, at least maybe 25 electrodes.
Yeah. So of course, the less electrodes, the less data you have, the less information you inject into the source mapping model. So the resolution will be much less, and therefore, depending on your expectations, regarding precise source mapping. Again, the rule of thumb is that the less electrodes you go, the lesser the resolution. So if your question is whether, this spike is coming, or this, epileptogenic form of a signal is coming more from the right frontal lobe than the left frontal lobe, or maybe ventral versus dorsal, maybe 25 electrodes might be good enough. But it's hard to give a very precise and definitive answer to that, because as you know better, every case is different. But from a purely modeling perspective, the less electrodes, the poorer the spatial resolution. So you have to lower your expectations, and the gain of moving from just scalp EEG to source modeling is actually less when you have less electrodes. But it's possible. If you ask whether it's legitimate and you can produce a source model with 25 electrodes, yes, actually you can even have a source model from 2 electrodes, but you understand that the predictive value of the source maps would be very, very poor with only 2 electrodes. And there are papers in the literature that actually quantify the degradation of spatial resolution as you reduce the number of electrodes. So there are some basic science papers that look into that with ground truth simulations, etc. But I think, even for basic research, but I guess it's also true in the reality of the clinic and clinical research in particular, I think you have to try. I know this is time consuming and a bit involved, but if you are curious, I would encourage you to try, these models on your own data and just see for yourself, whether you gain information or not. And maybe depending on what you're looking for and the degree of sophistication of the source mapping that you expect, then you can adjust, your expectations accordingly and continue to use source mapping or not. But this is definitely an experience you have to gain. That would be my recommendation.
Okay, thank you so much.
Thank you. I see in the chat that there is another related question to the digitization of electrode placement. And Carolina is suggesting that we look into maybe software that uses iPhone or smartphone cameras. And again, I'm not aware of probably the latest, greatest tools, especially with AI, in terms of image processing and recognition of objects in complex scenes. I'm sure that there could be, very interesting solutions there. I haven't seen anything reported of that would be robust, and especially with patients who may have a lot of hair. If you put an electrode cap, that's different, right? Because the hair is under the cap. So that's another reality. And then probably there are solutions we could use, and probably published solutions. I was more thinking of, the chronic recordings with glued electrodes directly on the scalp. This is problematic, but with a cap, there are probably solutions out there.
Yeah, thank you. Thank you, Raimundo and Sylvain. I have 2 questions. The first one will be related to the artifacts of the muscles that we have frequently during the seizures, especially at the onset of those tonic seizures. Because in the end, this is like a vector. So is there a way to minimize this noise so we can try to point it out when we are processing this and not getting a false positive or a false negative in terms of the seizure onset?
Yeah, no, that's a great question and a major issue, even in basic research with EEG or MEG. So this is really, really a confounding factor. I'm not sure I have a magic wand to remove these artifacts. I think it's what you could try to do is pre-process your data as much as possible using, for instance, ICA, independent component analysis, that basically takes all the channels at once and decompose the contents of the channels into statistically independent signal components. So on a good day, on a good patient, so to speak, in a good case, you would see maybe the muscle artifact captured by one component, and that would be distinct from the rest of the EEG. But, I think, you may expect the problem is not so much to confound muscle artifact with brain activity, because usually the muscle artifacts are very strong most of the time, right? So, whereas the high frequency activity of the brain in the same frequency range would be much smaller in amplitude. So the problem is more than that the muscle artifact just shadows and masks the brain activity in the same frequency range. So that's one major issue. So you can try ICA to, sort out what might be coming from muscles versus brain, because they would not be related in time. But that doesn't solve necessarily the problem of, muscle artifacts being much larger and just completely shadowing like gamma-like activity from the brain. So, but definitely doing a form of ICA, I think would be, would help actually sort out what would be the different components in your signal.
Okay, thank you. And the other question is, because we have been trying to use this on our own data, especially in focal cortical dysplasias, and to find a dipole is not as straightforward, since the activity is quite fast and small, and there's not like this easily recognizable pattern of a dipole. And since we don't have HD EEG as Jose remarked, what's your experience with this type of, because sometimes we don't have a proper image MRI, but we suspect there's something there, and we would like to add more information from the scalp EEG point of view to say, okay, maybe we do have a focal cortical dysplasia there. Do you have any tips on that?
Maybe my recommendation is that if you have any idea any idea, any conception of what would be the scalp manifestation of the FCD, you could basically mark them. So you can mark them using, for instance, brainstorm review tool, and you create like what we call events. So you would mark those things just like some of you may mark spikes, for instance.
Yeah.
And maybe one way to boost the signal level would be to look at all these events and average those events, just to create like an event-related potential, just to boost the signal level around those events and reduce the amount of ongoing brain activity or even noise around them. And therefore, you may be better equipped with, or you may have a better quality of a source model associated with the average of these events. And at least that would offer a first glimpse into, okay, where that might be generated, is it indeed frontal? Is it more temporal? Is it contained, within a region? Does it spread, etc.? Then what you could do is from this average define, so you do the source mapping of the average, then you use maybe the MRI template to project indeed the source, and then delineate a scout, as we call it, a region of interest, which would be circumscribed, or which would circumscribe the hotspot of that source. So you just take, I don't know, the 80% most activated cortical area at the peak of the average event or at the onset, you decide. And that's a region of interest from which you could extract ongoing brain activity as if it were a virtual electrode. But then you go back to the ongoing brain data, meaning, you know, each single event without before that you had before averaging. Then you look at the time series in that region, and does it systematically kind of reproduces this this kind of aberrant event that you saw in the average, just to verify the consistency of the activity in that region and whether this is indeed something that looks suspicious, right?
Yeah. Okay. Thank you.
No problem. Thank you.
I might maybe add two things regarding the high density EEG. It's of course used in some centers, and they do it mostly for research and sometimes for clinical evaluation using ESI. But there are some publications saying it might be suitable to use the 25 array electrodes. And yeah, I have a question. Is because is there a way to, for example, if I mark some spikes or some RPs, is it a way to try to automatically look in the rest of the recording, the ones that I selected? Is it possible using Brainstorm?
I'm trying to see how you could do that. So you could use the few that you have marked as a template, or even average the few that you have marked, and then that creates a template. I think, and Raimondo, please correct me or add the information, but there is a function in the Scout panel that lets you basically compute a form of correlation between a signal time series and your recordings. I would need to look into the details of that. I'm not even sure it's fully documented, but I remember this is something we had explored because I think this could be a very useful tool to add indeed. So this is somewhere in the Scout panel where you can again compute the correlation between, oh, that's probably a channel, and the rest of your EEG recording or the source time series. So you need to, must be a way to do that and to hack a little bit the brainstorm there. So this is a conversation we could continue on the forum.
Okay, yeah. I think
idea.
That's something very good, especially because that's an easy way to try to avoid going spike per spike, especially if you have, for example, two different sources or two different potential sources based on interictal epileptiform activity. So it's something that we can, thank you. And the other question is, how many spikes or how many RPs? Because of course, the more spikes, the better, but up to a certain point, probably, I don't know if you have 500 spikes, is it the same as having 100? But even in some patients detected 100 spikes is not that easy. For example, in patients who actually have, for example, infrequent interictal activity. Yeah. So this is something we've been struggling with, indeed, especially in MEG, because we see patients that are under medication. So, you know, as you know better than me, the spike rate, so to speak, might be altered by medication, etc. And we can only see them for like an hour or two. So it's very limited. In EEG, maybe the recording time can be longer. You can maybe modulate the medication, etc. So how many spikes, how many trials, basically repetitions of the event? There is no closed answer, right? Because it really depends where the spikes are generated. Is it deeper regions? Are they more superficial? Does it involve a lot of cortex, less cortex? Because it will condition the signal to noise. Because what is a spike at the end of the day? It's just something that emerges from ongoing brain fluctuations, right? So, and these fluctuations are not noise, but with respect to your interest of looking at spike generation, they are
possibly confounding signals. So you need to, for instance, average those spikes together. How many do you need? Again, it really depends on every patient. That's one of the difficulties, depending on where the spike is coming from and how strong the generators are, how far they are from the electrodes. So the deeper they are, the lesser the signal strength, and therefore probably more evidence needs to be accumulated in the form of more events. And it's the same in basic research, like in vision or audition, etc. We always wonder how many trials we need to come up with in the design of a study. And my answer is always very practical and pragmatic, which means, okay, we need to pilot this and maybe go for as many trials as we can shove into 1 hour of recording. But this you cannot do with a patient because, again, the nature of the signal is different and you cannot just ask the patient to produce small spikes for the sake of piloting your pipeline. So that's why also a lot of the research, I think, is not moving away from spike wave analysis, but is looking at ongoing brain activity with the assumption that an epileptic brain is always epileptic, even when it does not produce a spike and let alone a seizure. And I think it's a reasonable assumption, but the mechanisms probably are not well understood and probably vary from one type of epilepsy to the other. But that's one major interest of ours in terms of research, which is, again, looking at a patient as a whole, the whole hour of recording also, and process their resting state activity, ongoing activity, and looking at spectral, manifestations and aberrations with respect to normative data from the same age group and biological sex, etc. So this is where also maybe the future lies, by comparing a patient to normative data collected from healthy controls. And there are more and more of these data repositories, as you know. There's still progress to be made, but I think the science of epilepsy and the science of electrophysiology is going in that direction, which is very exciting because then the approach would be completely different and not necessarily rely on singular events. So I'm sorry, my answer is not helping much, but I think there is definitely hope with future approaches in that respect.
We gotta keep trying to collect as many as we can.
Yes, I guess.
Okay.
And how is it compared to the connectivity? I mean, is it the spikes we have found in certain locations is related to the difference in connectivity and the ongoing brain activity? I mean, have you measured that to see, okay, if we have this different type of connectome, let's call it like that, this might indicate that the activity, we might expect that the spikes might come from there?
So we haven't published in that respect ourselves, but I know that colleagues out there have looked at connectivity as a marker of epileptogenicity, for sure. Not only in electrophysiology, but in imaging in general, functional MRI in particular. And I think this is a very exciting development, right? To look at epilepsy as a network disease. And maybe spikes are, probably generated locally, but maybe are the outcome of complex interactions in larger networks, and with struggling, with areas that tend to be more irritated than others, excitable than others, and a confrontation with inhibitory signals coming from maybe the rest of the brain, including subcortical regions. So this is a big question mark here, and this is very exciting research that's going on. I don't think this is mature yet to be useful in the clinic, especially with low density EEG, because resolving connectivity measures is not fully. I mean, there are many ways of approaching this objective. There is no gold standard way to measure interactions in electrophysiology, as you know. There is coherence, certain forms of correlation, certain forms of phase locking between different regions, different frequencies. The mechanisms are not clear yet. But I think the idea is really interesting to pursue, to see these even, the generation of interictal activity as the fruit of network interactions. That's certainly the future as well.
Thank you.
Okay.
Thank you so much for the opportunity. I hope
No.
you have time and interest in trying the software for yourself. And we're looking forward to your feedback, and we know that from your community, there's a lot of good ideas and suggestions to improve the tools. So please share those with us and we can have a conversation.